So I'll just introduce where I come from to start with. Daniel Bryson and myself set up the Northwest Embryonic Stem Cell Centre in the University of Manchester about 5 years ago now. And this centre is has clean labs a little bit like Michael has already described. And these clean laboratories were, and still are actually, the first in the Northwest, not the Northeast, but the Northwest, to derive human embryonic stem cells at current good manufacturing practice standards. So that is at a standard that would be suitable for using them in clinical therapy. And you have a similar centre here in Newcastle. And we hope that this will allow scientists to develop stem cell products at GMP for potential therapeutic use. And this just shows our clean rooms here in the Northwest Embryonic Stem Cell Centre here in our core technology facility in Manchester. So what are embryonic stem cells? Embryonic stem cells are cells specific to the early pre-implantation embryo. And everything I'm going to tell you about relates to human embryonic stem cells, is what we are interested in, but of course you can derive them in other species. And the original derivation of embryonic stem cells were, of course, from Evans and Kaufman in the mouse, and Martin Evans was awarded the Nobel Prize for that work. So these stem cells are derived at the blastocyst stage, and they're present here in the inner cell mass of the embryo, and this is the bit that in normal development would go on to form the trilaminar disc and indeed all the different tissues of the body. And they have two specific important characteristics. The first is that they're capable in vitro of unlimited self-renewal. That is, they can divide to give more stem cells and more or less continuously continue to divide to give stem cells under the right conditions in a culture dish. Whereas given other conditions, they may generate non-stem precursors that can give rise to differentiated cell types. And this leads to the second particularly important characteristic. Because they can generate these non-stem progenitors, they can generate such progenitors which can give rise to the differentiated cell types of the 3 embryonic germ layers. So again, Michael asked you to remember your embryology. Hopefully you might remember that in embryonic development there are 3 major tissue types, the ectoderm, the mesoderm and the endoderm, which give rise to the different categories of differentiated cells, ectoderm giving rise to the nervous system and skin and so on, mesoderm to cardiac muscle, kidney, red blood cells and skeletal tissues, and endoderm to the gut and its derivatives. So these 2 major characteristics, they're pluripotential, the potential to give rise to such a diverse range of cells, differentiated cells, and this unlimited self-renewal are particular characteristics that make these cells useful for a number of purposes. So in Manchester we have been, I'm going to stand up here so I can actually see this, we've been looking at generating embryonic stem cell lines from these stem cells. And we start off with the IVF clinic, where under our HFEA, Human Fertility and Embryology Authority license, we can accept embryos that are donated by couples attending for IVF or other ARTs that are surplus to their requirement for treatment or for donation to other couples. And under informed consent, these are donated to our research or stem cell program. And these embryos are then developed in vitro in the culture dish to the blastocyst stage at about day 5 to 6, where you can see the inner cell mass here. It's not as nice as the inner cell mass in a mouse embryo. This is actually a very nice human embryo. Some human embryos look pretty rubbish, actually. But those that do have nice inner cell masses, we can then use to generate embryonic stem cell lines. And at this stage, we've got about 30 to 120 cells. So these embryos are then plated onto a feeder layer, shown by these pink cells here. And these are inactivated fibroblasts. For research purposes, these would be mouse embryonic fibroblasts. But for our purposes, because we don't want any animal products, we want to have xeno-free system, we used human fibroblasts, which have actually been derived at GMP standard. So these embryos are plated whole or as isolated inner cell mass onto the fibroblasts, and then with time and given the right conditions, we generate cells which start to proliferate and then can be subcultured to give an established embryonic stem cell line. And if we want to use these cells for clinical purposes, we would then have to put them through some kind of differentiation protocol that I'm going to tell you a little bit about. So if we're going to use them therapeutically, as Michael's already nicely introduced, we're going to also need licensing under the European Tissue Act from the HTA, or in America from the FDA. So we have now derived 16 lines in Manchester. 10 of these are research grade, and 6 of these are clinical grade lines. And one of them is actually xeno-free. And this is done, as I said, under CGMP conditions, right from the IVF labs through to the generation of the cell line and storage in defined serum-free medium with CGMP human feeder cells, validated reagents, and with full paper trail, which is absolutely essential for doing anything for clinical purposes. And our clean rooms are licensed both by the MHRA and the HTA. And this just shows one of our lines and the process of its derivation. Day 4, cultured embryos, the embryo hatching from the zona pellucida here. Here's the embryo, and here's the inner cell mass. The inner cell mass-derived cells at passage 1, and here at passage 5, where you can see these nice colonies growing on these human feeders. So if we're going to tell you that these cells are stem cells, we've got to be able to show that they are in some way. And we need to characterize them. So we characterize our lines, as do others that are generating lines. And embryonic stem cells express particular marker proteins and carbohydrates indicative of their undifferentiated pluripotent state. Some of the carbohydrate antigens that are used are very well known as embryonic stem cell markers, as are shown here. We've got these nice colonies staining with these markers. But perhaps more important, there are 3 transcription factors, OCT4, SOX2, and NANOG, which, if you like, are key master transcription factors in a transcription factor network which regulates and maintains the ES cell state. And so we need to absolutely show that these cells are expressing these particular markers. So to grow human ES cells, we can maintain these as self-renewing, undifferentiated colonies for many generations, possibly indefinitely. And this probably contrasts with adult stem cells that in culture have a tendency to senesce. But to do this, we need, as I've just told you, to grow them on a feeder layer, which isn't very convenient and also is not defined, because these feeder cells are making lots of goodies, but we never know if they're making the same goodies from batch to batch, and we don't know what all those goodies are. So the alternative is to develop a system which doesn't require the feeders. And there are now proprietary feeder-free medium that you can use for growing human ES cells. And we have to add growth factors to these, and we have to grow the cells on an appropriate substrate. Extracellular matrix substrate, such as Matrigel, again, not good, from an EHS tumor, undefined, or specific recombinant proteins. So I'm not going to go into detail about how these cells are regulated, but if we want to use cells to push them to particular differentiated cell types to use for any purpose, for clinical therapy or for other in vitro purposes, we have to understand how they're regulated, because if we don't keep them in the stem cell state in a very controlled way, when we start to differentiate them, we'll get a complete mess. We'll get a whole mixture of different differentiated cell types, because we'll have some cells which actually weren't stem cells in our mix at the beginning. So we have spent a long time studying what, as many other groups, what regulates these cells. And I already mentioned that this transcription factor network, which is absolutely critical to maintaining this self-renewing pluripotent cell type, but also modifiers of the epigenetic status are also important. And recently, small RNAs that regulate the translation or degradation of messenger RNA within the cells are also shown to be very important. As well as these factors inside the cell, there are also external factors. Factors such as, very well known for mouse ES cells, leukemia inhibitory factor that you have to add to the medium to keep these cells as stem cells. And in for human ES cells, FGF2 and TGF-beta family molecules have been shown to be critical. And we still don't thoroughly understand the role of the matrix, but we know these cells like to grow attached to a matrix substrate. And the third characteristic here that is very important is the oxygen level. Some studies have shown that growing these cells at low oxygen is very beneficial for maintaining them as stem cells. So if we know what these conditions are, then we can devise these kind of defined systems. Because if we want to use cells for transplantation for any reproducible purpose, we need a defined system. And so in Manchester, we developed a defined feeder-free system. I said there are now proprietary systems you can use. And if you do this, you can grow these cells now in a feeder-free manner. You can see these nice small cells, characteristic. They have a very characteristic morphology, small cells with a very prominent nucleus and a very prominent nucleolus. You can grow these cells in a culture dish, and they will just expand over about 3 days to fill the dish, and you can work out the growth kinetics as shown down here. So how do I know that these cells can differentiate into all these different cell types? Well, we and also others have put these cells into immunocompromised mice. And if you do this, you put them under the kidney capsule or subcutaneously, then embryonic stem cells, human embryonic stem cells, will develop to form differentiated tumours containing derivatives of all those 3 germ layers, indicating their pluripotency. So there's sort of some neuroepithelium here, bone, cartilage, mucosal cells, smooth muscle, and some primitive tubules similar to kidney tubules, suggesting that these cells really are, as I've indicated to you, able to give rise to all these cell types as they would normally in the body. Okay, so we can derive these cells, we can grow them in culture, we can show that they have this potential. What can we do with them? So one thing that we're very interested in, because we're using human ES cells, is that we can understand normal human development and what goes wrong in congenital defects. And we can understand this in a dish by growing these cells in vitro and pushing them to different differentiated cell types, and then studying them in a way that we couldn't possibly do in human. We can't do the sort of mouse studies that people have done, making transgenics and so on. Of course, what the media has latched onto is the potential of these cells to be directed to give specific cell types in culture which could be used for cell therapy. So bone marrow for leukemia, nerve cells for Parkinson's disease, heart muscle, pancreatic beta cells, and so on. And what the benefit of human embryonic stem cells here is that you could expand them in culture to make large numbers of cells. But then you've still got the challenge of directing their differentiation specifically to the particular cell type that you want before you transfer them back to replace heart muscle for heart disease, pancreatic beta cells in a patient with type 1 diabetes. And I'll come back to that. So the other form of pluripotent stem cell, called induced pluripotent stem cells, which are actually somatic cells which have been reprogrammed to an ES-like state, may be very useful for this area. So pharmaceutical industry have really lacked models for human cell models, in vitro cell models, for drug screening, for drug discovery, and for toxicity testing. And now we have human cells that we can push into these different differentiated cell types, and we can use them to test different drugs outside the body. And these are human cells instead of the rather poor human models or mouse cells that they use at the moment. Okay, so we've got to find a way to push these cells in the direction that we want to, and this is, any of the developmental biologists in the audience will realize that this is the Waddington developmental landscape. It's just to illustrate that what we're trying to do here is that we're going from a pluripotent stem cell state here, and we can imagine the progress to differentiation as rolling a ball down a hill in which there are a number of valleys. And these valleys lead to different terminally differentiated cell types. And different inducers and triggers will push these cells to go down one valley or another. But along the way, there are these stable states, such as the pluripotent stem cell state here, and possibly here, a state that might represent where adult stem cells are. So what we're going to do is try and push these cells in a very controlled way down one of these valleys to a differentiated cell type, and that alone. So how can we do that? Well, the conventional way has been to aggregate these cells in suspension to form embryoid bodies, effectively an aggregator of ES cells. And if you do this in the presence of serum or defined factors, then you get differentiation, but this is generally heterogeneous and uncontrolled. So an alternative way to do it is to co-culture the cells with particular inducing factors that may be able to control differentiation. But again, this is like putting the cells as stem cells with a feeder layer. It's undefined. We don't really know what we're doing, and it's very difficult to get it reproducible. So what would be a better way would be to remove them from feeder cells and add particular growth factors, hormones, and other factors. And this has been done by a number of groups. We can add these in a systematic and sequential manner to direct differentiation along to particular desired developmental pathways. But the downside here is the efficiency may be low. And we may need to select for the particular cells that we want. We may not get all our cells to go to cardiomyocytes or hepatocytes, whatever. So this is just showing the embryoid body route. These are embryoid bodies, ES cell aggregates in vitro, which to some extent mimic the early stages of development. And it's believed that the cell interactions in these embryoid bodies make differentiation more efficient than in a 2D culture dish. And if you do this kind of work and then you plate those embryoid bodies onto a culture dish, then you do indeed get terminally differentiated cell types, neural cells shown here, endodermal derivatives, and mesodermal derivatives. But the problem is you get them in a sort of hodgepodge, in a whole mixture of different cell types, and it's completely impossible to control. So what people have tried to do is to go and look at what happens during normal development and push the cells down particular lineages. So for instance, if you wanted heart muscle, you would look at what happens at each of these different stages in development, generally extrapolating from data on the mouse, and then look at the factors that have induced each single stage to the next and try and recapitulate that in a culture dish. And at present, no study has really shown definitively that ES cells can give 100% of a desired targeted cell type. But there are very good enrichment protocols here using combinations of exogenous growth factors. So one example I'm going to give you is for getting human ES cells to form pancreatic beta cells. So we start with an ES up here. We would go through an endodermal precursor, and then we would get some kind of pancreatic precursor. Can't put it really back, if you're thinking about cell therapy, into the pancreas, which is going to be completely shot in type 1 patients. So it will probably be put back into the liver in the same way that pancreatic transplants are. And we know quite a lot about how to get from an embryonic cell to a pancreatic precursor. Some of the factors are shown here. And so DeMoore and colleagues and Doug Melton's group some years ago now developed this 5-stage protocol using a sequence of combinations of growth factors and other compounds to go effectively from ES and recapitulate development, going through all these developmental stages to give rise to endocrine beta cells. But the early protocols were really very poor. They gave only about 12% insulin-secreting cells, and these cells were immature. They could respond by secreting insulin to compounds, molecules like KCl, but they couldn't respond to glucose, which is pretty hopeless since that's the only real thing that you really want them to do. But more recently, there are improved protocols, and these have given cells which, when put back into mouse models of type 1 diabetes, can rescue glucose responsiveness. And indeed, with refined protocols using small molecules to manipulate signaling pathways, we can actually get about 50% pancreatic precursors in the culture dish. So this is quite promising, but challenging. So we took the approach of looking at generating chondrocytes from human ES cells. And at the time we started this, several studies had reported some success in generating chondrogenic cells from human ES cells using this embryoid body technique. But we decided to take a different approach and to try and use a completely defined system and to recapitulate development. And it was based on our original feeder-free system. And this initial work was done by Rachel Aldershaw, who's now here in Newcastle when she was in my lab. So we used a 2D chemically defined culture system. And the rationale here was that we could then allow maximum exposure of the human ES cells to these different compounds that we put into the culture dish, and hopefully therefore push them all down the chondrogenic lineage. And why chondrocytes? Well, osteoarthritis is the most common form of arthritis, one of the greatest causes of disability in the UK. It's an age-related disease. I know you have a lot of activity and work, as Michael's already alluded to, here in Newcastle. And it affects a large number of people, and as the population ages and with increasing obesity, this is probably likely to increase further. It also costs the NHS a lot of money. And we also have the possibility of using chondrogenic cells for patients, younger patients with sports injuries, patients where prosthesis would not be appropriate because the joint or hip replacement is going to have a much shorter lifespan than that of the patient, and where currently autologous cartilage grafts are being used. So we tried to recapitulate development in a 3-stage process, going from ES cells to a mesendoderm type cell type, to a mesoderm cell, and then to chondrocytes. Meanwhile, avoiding all these other lineages that we didn't want. And I won't go into detail here, but basically we developed a series of growth factors that we used sequentially in these different stages to block the unwanted lineages and to push these cells towards chondrocytes. And the end result is showed here. At the end of stage 3 of our protocol, we have these aggregates of safran O positive cells indicating the presence of chondroitin sulfate proteoglycan, the characteristic proteoglycan of chondrocytes. Importantly, this protocol was highly efficient. Depending on the cell line we used, we had between 75% and 97% of these SOX9 positive cells, SOX9 here being the critical transcription factor which regulates COL2 production, the major matrix molecule of cartilage. And we've gone on from this now, because it's one thing developing cells in culture, to do preclinical trials. I shouldn't call them trials, should I? Preclinical research. And we've now started to put these cells into animals, because the question is, can they actually make cartilage? So we've used a thymic nude rats, and we've made osteochondral surgical defects in these rats, and show that if we do that, then we can indeed get cartilage repair. So these are our controls here. You can see these nice sapho stained cartilage in the knee joint. This is our defect control, where we put no cells in, complete lack of cartilage. And this is what happens when we put our ES-derived chondrogenic cells in. You can see this nice cartilage repair. It's not perfect, but it's definitely very encouraging. And we've now done a similar protocol with induced pluripotent cells that Lyle is going to tell you about. So just finally, one other example is where we think there is a lot of potential for cell therapy, cardiomyocytes. So using, using a number of protocols, it's been possible to show that you can get beating muscle from ES cells in culture. And Christine Mummery's lab pioneered co-culture of human ES cells with enoderm cell lines to induce high levels of cardiomyocyte formation. But again, using co-culture is not ideal, and I'm going to show you that there are some more developed methods just in the last but one slide. Using these cells, people have grafted them in preclinical work into the hearts of mice, pigs, and guinea pigs, and shown that these kind of cells can take on pacemaker function or improve heart rate in experimental infarct situations. But the problem is, this tends to only be in the short term. And so I think at the moment, we're still in the experimental stages in terms of using ES cells for cardiomyocyte repair. So more recently, people have tried to do the same kind of approach I've told you before, have tried to recapitulate development and get heart muscle by using a number of factors in the culture system, which has been quite successful. And this recent paper by Yamazaki et al. Used beating clusters derived from human ES cells in a high throughput screening system to look for drugs that influenced the QT interval between polarization and depolarization in the heart rhythm. And so what they're using this for is to look for drugs that would change the heart rhythm. And this is a major need for toxicology studies for the pharmaceutical industry, because many drugs have a side effect of influencing the heart rhythm, and just illustrates this use of ES cells in toxicity research. And the top toxicology targets are probably cardiomyocytes, hepatocytes, and kidney cells in biopharma. But the other two cell types, although being used, are perhaps slightly less advanced. Okay, so finally, have we got to any clinical trials using ES cells? Some of you may be aware that in 2010, Geron announced a multi-center phase 1 clinical trial for using ES-derived oligodendrocytes for spinal cord injury patients. And this was a safety trial. The idea was they had a protocol to derive oligodendrocytes from these ES cells, and then these would be used to see if they were safe when grafted into the spinal cord of patients that, where really all other remedies had been trialed. And indeed, the few patients that went into this trial, the cells seemed to be quite safe. They didn't develop tumours. However, unfortunately, the fact that maybe we will say their patients did not get up and walk immediately after the injection of these oligodendrocytes caused some considerable damage to the share price of Geron. And the difficulties of raising finance for these kind of trials meant that in November 2011, Geron pulled out of this pluripotent stem cell therapy field and is now concentrating on cancer. But I would hasten to say it wasn't because the trial failed, it was an issue of the financial climate. And of course, I think Michael has already alluded to the work that's going on in London on using ES cells for repair of the macula in patients with macular degeneration. In particular, it looks most promising for Stargardt's macular degeneration. So adult stem cells have not, I understand, been very promising here, but retinal pigment cells obtained from ES cells have actually been used in this trial by this company, Advanced Cell Therapies. And this original safety trial, these cells at least have not caused tumours, and there is some apparent slight visual improvement in one of the patients. So this route here may be a promising one, particularly because the eye being at least a semi or fully immunoprivileged site would allow cells that are not necessarily the same tissue type of as the patient to be used. So my final slide is just showing here as a sort of sum up therapeutic potential, ES cells and adult stem cells. So ES cells are pluripotent. They can differentiate in more cell types than AS cells. Adult stem cells tend to have a more restricted but not restricted differentiation potential. In culture, ES cells can be expanded, so they can be grown up into large numbers over a long period of time. It may be possible to make ES cell banks. And ES cells, it's probably more difficult to expand these. However, we've got the problem of immune rejection for ES cells in cell therapy, meaning that unless we have an immunoprivileged site, we would need to tissue type and match these cells. For adult stem cells, there's not going to be a rejection problem if the patient's own cells can be used, and that's not always possible. And indeed, some adult stem cells, particularly MSCs, have an immunosuppressive effect that can be useful, although in some patients might be detrimental if that patient is immunocompromised. Surgery, 1 operation. Surgery, 2 operations if autologous. Ethical considerations. ES cells are considered by some to be controversial. AS would not be controversial, particularly if the patient is donating their own stem cells or from a consenting adult donor. And finally, safety. We have to consider that ES cells can make those teratomas I told you about, and there's got to be exhaustive trials to show that there are no ES cells remaining when you graft in any differentiated cells from the original cell population. That was done in the ACT macular trial. For AS cells, cancer is less likely, although there are some reports of inappropriate differentiation when MSCs are used, and also these cells do migrate, so they might go elsewhere in the body. So finally, just like to acknowledge my own group, to think the work that came from our lab, which certainly wasn't all of it, Those people in red contributed to the chondrogenic work. The people in blue down here have contributed to our GMP derivation of ES cells, and I've got a number of collaborators. So thank you for your attention, and if anybody wants to ask any questions, please do.